Hello friends, welcome to Sam Biology class. Gene interaction takes place when genes at multiple loci determine a single phenotype. There is two types of gene interactions. Number one, interaction that produce novel phenotypes, and number two, interaction with epistasis. First one is gene interaction that result in novel phenotypes. In this case, genes at two loci interact to produce a single characteristics. Take the example of summer squash, scientifically known as Cucurbita pepo. In summer squash, fruit shape is determined by two genes A and B. Each gene has two alleles. One is dominant and the other one is recessive. When both genes have at least one dominant allele, then the fruits are disc-shaped. So this is the genotype that produce disc-shaped fruits. But when only one gene has dominant allele and the other gene has only recessive allele, then the fruits are spherical. These are the two genotypes that produce spherical fruits. But when both genes A and B have only recessive alleles, then the fruits are elongated. So in Cucurbita pepo, two genes A and B interact to produce a single characteristic, which is the fruit shape. In this example, spherical fruits are novel phenotypes which are produced due to the interaction between two genes. Second type of gene interaction is epistasis, in which one gene masks or hides the effect of another gene at a different locus. The gene that does the masking is called epistatic gene, and the gene whose effect is masked is called hypostatic gene. There are different types of epistasis. First one is recessive epistasis. Which is seen in the genes that determine coat color in Labrador retrievers. In these dogs, the coat color can be black, brown, or yellow. These coat colors are determined by interaction between genes at two separate loci. Locus 1 has the dominant allele that encode black pigment and its recessive allele brown. In the second locus, the dominant allele allows the deposition of dark pigments, black or brown, in the hair shaft, and its recessive allele prevent the deposition of pigments. As a result, coat become yellow. Black pigments are produced when first locus has at least one dominant allele and the second locus also has at least one dominant allele. So this is the genotype that result in black coat. Black coat. But when the first locus has only recessive allele and the second locus has at least one dominant allele, this result in brown coat. This result in brown coat. But when first locus has dominant alleles and second locus has only recessive alleles, then the deposition of black pigment is prevented. As a result, coat become yellow. Similarly, when both genes have only recessive allele, then also the phenotype is yellow coat. Phenotype is yellow coat. In this example, the recessive allele of one locus is epistatic to both the alleles of the second locus. So the alleles for black and brown pigment are hypostatic. Such type of interaction is known as recessive epistasis because the recessive allele when present in homozygous state prevent the expression of both the alleles of the other locus. Second one is dominant epistasis, in which a single copy of a dominant allele is required to inhibit the expression of the alleles at a different locus. Dominant epistasis is seen in the interaction between two loci that determine fruit color in summer squash. The dominant allele of the first locus inhibit the production of pigments. The dominant allele inhibit the production of pigments. As a result, fruits become white. Its recessive allele, when present in homozygous state, allow pigment production. As a result, fruits become color. Second locus determine the type of pigments produced. Its dominant allele encode yellow pigments. The dominant allele encode yellow pigment and its recessive allele when present in homozygous state encode green pigment. These are the genotypes that determine different fruit colors in summer squash. In both first and second genotype, the first locus has dominant alleles that inhibit the production of pigments. As a result, white squash are produced. In the third genotype, the second locus has dominant allele that encode yellow pigments. Presence of only recessive alleles at the first locus allow the production of yellow pigments. As a result, squash become yellow. In the fourth genotype, presence of only recessive alleles at both loci allow the production of green pigments. As a result, green squash are produced. In this example, the dominant allele of the first locus is epistatic to both the alleles of the second locus. So the alleles of the second locus are hypostatic. Last one is duplicate recessive epistasis, in which the presence of two recessive alleles at either of the loci are capable of suppressing a phenotype. Such type of interaction is illustrated by albinism in snail. Albinism is the absence of pigments. Albinism is the absence of pigments. It is a common genetic trait in many plants and animals. Almost always, pigments are produced by multi-step biochemical pathways. Multi-step biochemical pathway. Albinism thus involves gene interaction. These are the different genotypes. In the first genotype, due to the presence of dominant allele at both loci, pigments are produced as a result of which the snails become pigmented. Presence of two recessive alleles at the first locus of second genotype and at the second locus of third genotype suppress the expression of phenotype and snails become albino. In the last genotype, both loci have only recessive alleles. This leads to the absence of pigments. As a result, albino snails are produced. So friends, this is all about different types of gene interaction. For more updates, please subscribe my YouTube channel, Sam Biology Class.